The lymphatic system is a vast network of vessels running through the body. It has a number of functions, including elimination of water that congests tissues. Every day, blood circulation releases large amounts of liquid into the body's tissues. The vessels in the lymphatic system collect this liquid and keep tissues from swelling. The liquid, called lymph, circulates in one direction in the lymphatic vessels. On its way, the lymph passes through small clusters of organs along the vessels, the lymph nodes. These nodes contain many of the body's defense cells, with which they filter the lymph to eliminate infectious agents. Once it is cleansed, the lymph is returned to the blood circulatory system via the subclavian veins. Every day, the lymphatic system drains and cleanses about 3 liters of lymph. The air around us contains all sorts of bacteria and viruses that can attack our body and cause diseases. Immunity consists of all the defense mechanisms the body uses to fend off these infections. The skin forms a physical barrier against disease-causing microorganisms. If microorganisms manage to breach this first defense, they enter the body and attack its cells. When a cell is infected, a series of defense mechanisms is activated. This is called the inflammatory response. Specialized cells, the white blood cells, are sent to the site of the infection. Some white blood cells are capable of surrounding and digesting the foreign bodies. This process is called phagocytosis. Phagocytosis is not always sufficient to eliminate infection. In this case, other white blood cells intervene by producing antibodies. Antibodies are proteins that attach themselves to infectious microorganisms and help to destroy them. If none of these mechanisms gets rid of the infection, medications may be used to back up the immune system.
The immune system is the body's defense against foreign materials and pathogens. It uses the 3 lines of defense to respond to invasion. A major part of the immune system is the lymphatic system, which has a circulatory and an immune function. It contains a network of vessels, nodes, tissues, and organs. This network makes and transports fluid called lymph, which contains lymphocytes. B cells and T cells, 2 specialized kinds of lymphocyte, are produced in the bone marrow. Lymph nodes destroy pathogens by filtering foreign particles and trapping them in fibers. The immune system has many components, comprising its 3 lines of defense. The first line of defense includes skin, mucous membranes, natural secretions, and the body's natural flora. The second line of defense involves protein-based responses and cell-based responses. The first 2 lines of defense are non-specific. They provide a barrier to any pathogen, bacteria, virus, or protozoa. But the identity of the pathogen is crucial for the third line of defense. The third line of defense involves B cells and T cells that identify and try to destroy the pathogen. The body can identify foreign material and pathogens because it can distinguish between matter that is self and non-self. For example, people have different blood types caused by red blood cells with specific protein markers on their surface. Someone with an A protein, type A, cannot receive blood from someone with a B protein because the body identifies the B protein as non-self. All other cells in the body have a different set of genetic protein markers that are part of a major histocompatibility complex, MHC, which is unique to you. B cells and T cells recognize their own MHC markers, self, but will attack material with foreign MHC markers, non-self. If a pathogen with a different MHC breaches the first two lines of defense, the immune system realizes something is not right and turns to the third line of defense. This also applies to tissue and organ transplants. If the MHC of an organ donor and a recipient is not closely matched, it triggers an immune response that rejects the transplanted material. The first line of defense is always on guard and is effective against most pathogens. It includes skin, mucous membranes, natural secretions, and natural flora. If any of these components are not working properly, a pathogen could breach the defenses and cause an opportunistic infection. Healthy skin is the major component and can prevent the entry of most viruses, bacteria, and other infections. The epidermis contains cells full of the protein keratin, which makes them tough and impenetrable by water and microorganisms. If the skin is cut, clotting prevents blood loss and helps wounds heal fast. Salt contained in sweat and fatty acids secreted by glands restrict the growth of fungi and bacteria. Mucous membranes line the respiratory, urinary, gastrointestinal, and reproductive tracts. The membranes create mucus that traps pathogens. Cilia move pathogens to where they can be expelled, for example the throat, where coughing or sneezing can expel pathogens from the respiratory system. Natural secretions include saliva, which contains antibacterial agents that can kill some bacteria, stomach acid, which kills many bacteria ingested with food, and tears, which contain lysozyme, the enzyme in lysosomes that can digest bacterial walls. Natural flora are the body's own non-pathogenic bacteria. They limit the space available for pathogens. They occur throughout the body, but particularly on the skin, in the intestine, and in the vagina. Opportunistic infection may occur if some of this natural flora has been killed, for example by antibiotics, allowing pathogenic organisms to move in. The first line of defense is non-specific, operating no matter what pathogen is encountered or whether it has been encountered before. If the first line of defense is breached, the second line of defense has two types of response: protein-based and cell-based. In this chapter, we will look at protein-based responses. Proteins in the second line of defense include cytokines, interferons, and complement proteins. When bacteria attach themselves to a cell and try to push through its membrane, the cell's cytokines signal to neighboring cells that it is being attacked. Interferons are a subclass of cytokine that are secreted when a virus invades certain cells. Interferons trigger the production of antiviral proteins in neighboring cells and make them more resistant to the virus. Interferons are particularly important when a virus does not have far to travel. For example, cold viruses infecting nose or throat cells, and there is little time for the body to create antibodies. Complement proteins are blood proteins that assist the second line of defense in several ways. Some complement proteins stick to invading bacteria. Others stimulate the production of cells called phagocytes, which engulf and destroy bacteria and attract them to the infection site. Others destroy the membranes of invading microorganisms. If the first line of defense is breached, the second line of defense has two types of response: protein-based and cell-based. In this chapter, we will look at cell-based responses. Cell-based responses in the second line of defense include natural killer cells, phagocytes, inflammation, clotting, and fever. Natural killer NK cells recognize distress signals on the surface of an infected cell. Once the tumor or virus-infected cell is identified, NK cells kill it using cytotoxins. Phagocytes are a group of white blood cells that engulf and eat other cells. The process of phagocytosis begins with a macrophage detecting microbes by chemitaxis and sticking the microbes to its surface. It engulfs the microbes with its pseudopodia, flexible cell extensions. It then encloses the microbes in a vesicle, membrane-encased sac, called a phagosome, in which the microbe is eventually digested. Phagocytes include eosinophils, neutrophils, and monocytes. Eosinophils discharge enzymes to fight pathogens. Neutrophils, the most common white blood cell, are the first immune cells to arrive at an infection site. They do not survive phagocytosis. Monocytes are the largest white blood cells and become macrophages when they leave the bloodstream. Monocytes produce cytokines and can trigger a third-line response when T cells detect tiny fragments of foreign matter left in them after phagocytosis. If we have a cut or wound, the area may swell, turn red, develop pus, and become painful. Inflammation aims to destroy the infection, limit its effects, and replace and repair damaged tissue. Redness and swelling is caused by dilation of blood vessels. This increases blood flow to the area, bringing phagocytes. Phagocytes and damaged cells release histamines, which attract even more phagocytic cells to the infection. Phagocytes and proteins squeeze into infected tissue, causing swelling and pain. Because the affected area hurts, you're more likely to rest it. Pain is a sign to take things easy, so you have more energy to fight the infection. Pus contains dead white blood cells, body fluids, and damaged tissue, and also live white blood cells continuing the fight. Pus is reabsorbed by other cells or released. Blood clotting prevents further blood loss from an injury. Platelets help to form blood clots, prevent pathogens from entering, and repair wounds. When fighting an infection, we may get a fever, a high temperature with cold, clammy skin, and the shivers. Later, we may sweat a lot, meaning the fever has broken. A fever occurs when macrophages, fighting infection, secrete cytokines called interleukins. Interleukins help reset the hypothalamus, the body's thermostat, above 38 degrees Celsius or 100 degrees Fahrenheit. Despite the higher temperature, our skin may feel cold. This is caused by blood vessels being constricted, preventing heat loss. Shivering helps increase the temperature further. High temperature causes drowsiness and lowers body activity, allowing more energy to be spent fighting the pathogen. It also increases metabolism and enzyme activity to speed up the fight, while also preventing many pathogens from functioning properly. Recovery is usually signalled by the hypothalamus resetting to its normal 37 degrees Celsius. Sweating follows to cool the body down. The second line of defence is non-specific, operating no matter what pathogen is found or whether it has been found before. There are two types of response in the third line of defence: cell-based immunity and humoral, antibody-mediated immunity. In this chapter, we will look at cell-based responses. The third line of defence is specific, unlike the first and second lines, which are non-specific. Non-specific means the immune system reacts the same way to all infections, has no memory of previous infections, and gives the same level of response for each infection by the same organism. For example, food poisoning bacteria like E. coli must invade the first two lines of defense each time you eat infected food. Specific means the immune system reacts in a specific way to an infection, has memory of previous infections, and gives a greater response for each subsequent infection by the same organism. For example, a childhood bout of measles or chickenpox creates lifelong immunity because the body remembers the pathogen and increases its defensive response to quickly subdue it if it reappears. Major component of cell-mediated immunity. Like all lymphocytes, T cells are produced in the bone marrow, but they mature in the thymus gland. The T stands for thymus-dependent. T cells recognize and fight against bacteria and viruses that live in host cells, fungi, worms and parasitic protozoa, cancerous cells, and foreign cells such as transplanted tissue. T cells recognize antigens in both infected cells and macrophages. There are two main types of T cell: T helper cells, TH, and cytotoxic T cells, TC. T helper cells recognize antigens in macrophages that retain some of the foreign material they ingested in the phagocytic process. T helper cells stimulate the formation of B cells, which secrete antibodies. When T helper cells are disabled, such as by HIV, sufferers often die of opportunistic infections that the body cannot fight off. Cytotoxic T cells identify cells infected with an antigen and kill them before they can replicate. They contain the protein perforin, which punches holes in the target cell's membrane. There are two types of response in the third line of defense: cell-based immunity and humoral, antibody-mediated immunity. In this chapter, we will look at humoral immunity. Like cell-based immunity, humoral immunity is a specific response. B cells are produced from stem cells in the bone marrow. Each B cell can recognize and fight one type of antigen only. Those antigens are the bacteria, for example tuberculosis, and viruses, such as influenza, that live outside cells, as well as toxins produced by bacteria. B cells have an immunoglobulin, Ig, or antibody on their surface. But how does a B cell find the only kind of antigen it is able to attack? In 1955, Sir Frank Macfarlane Burnet developed the clonal selection theory to explain this. B cells' antibodies act as receptors. When they find the antigen that matches them, they bind with it. The B cell then reproduces rapidly, forming a large clone of identical cells. This process is called clonal expansion. Many of the cells differentiate into plasma cells, which make large quantities of the same antibody to fight the antigen. Plasma cells can produce 10,000 molecules of antibody per second and live for up to a month. Antibodies coat the antigens and allow second-line defenses, such as phagocytic cells and complement proteins, to operate. Other cloned cells become memory B cells that know how to make this same antibody. They can survive for years or a whole lifetime. Memory B cells react so fast and vigorously to another infection by the same antigen that there is no time for the body to develop symptoms. The body is considered immune to the antigen. There are several classes of antibody with a common core structure, a constant and variable region. The constant region is relatively unchanged between antibody types. The variable region uses a bank of genes that are transcribed, translated, and spliced together in one of an estimated 20 billion combinations. The antibody immunoglobulin G, or IgG, activates complement proteins and directly neutralizes toxins. When IgG binds to its antigen, it changes shape to signal this, which recruits more IgG molecules and activates complement proteins. When the body recognizes an infection, antibody levels rise dramatically. But as the infection is fought off, antibody levels decrease until the next time the pathogen tries to enter the body. Non-specific, treating all pathogens the same way. The second line of defense responds if the first line has been breached. It is also non-specific, and it consists of protein-based responses and cell-based responses. There is some interaction with the third line of defense. Protein-based responses include cytokines, interferons, and complement proteins. Cell-based responses include natural killer cells, phagocytes, neutrophils, monocytes, inflammation, clotting, and fever. The third line of defense is specific, identifying the pathogen it is fighting against and remembering it for any future recurrences. It consists of cell-based immunity and humoral or antibody-mediated immunity. Cell-based immunity involves T cells, including T helpers and cytotoxic T cells. Humoral immunity involves B cells and producing antibodies to detect and bind antigens.